This program is for Bachelor of Science third year students, third year BSc, subject Botany, semester 6, course code is BOC 108, course title Cytogenetics and Plant Breeding. The title of the unit is Gene Mutations, Unit 7, Molecular Basis of Mutations is the module name today. I'm Ms. Lynette Fernandez, Associate Professor of PES's Ravi S. Naik College of Arts and Science, Farmagudi, Ponda, Goa. This module deals with tautomerism and frameshift mutations. In this module, we will be talking about the mechanisms by which tautomeric shifts in the bases in DNA causes mutations. It also explains how insertions or deletions can cause frameshift mutations. Gene mutations at the molecular level are permanent alterations in sequences of nucleotide bases in the nucleic acids which forms the genetic material. These alterations in the base sequences may be due to deletion of bases, insertion of bases, inversion of the sequence of bases, or even substitution of base pair. Deletions, insertions, inversions, these include those changes in the base sequences which involve breakage and reunion of DNA segments. However, in the case of substitution of base pair, here the base pair substitution may take place during the replication of DNA without the breakage of DNA. Base pair substitutions can be of two types, transitions and transversions. In transitions, a purine is replaced by another purine base. That is, adenine might be replaced by guanine, or guanine may be replaced by adenine. A pyrimidine is replaced by another pyrimidine. So, thymine might be replaced by cytosine, and cytosine might be replaced by thymine. The other type of base pair substitution is transversions. In transversions, a purine is replaced by a pyrimidine and vice versa. So, adenine can be replaced by cytosine and vice versa, and thymine can be replaced by guanine and vice versa. There are three substitutions as is shown in the figure. 1 transition and 2 transversions possible for every base pair. A total of 4 different transitions and 8 different transversions are possible. Transitions are very common. They are more common as compared to transversions, and often tautomeric shifts are responsible for transitions. What is tautomerism? The ability of a molecule to exist in more than one chemical form is called tautomerism. Transitions can be produced by tautomeric shifts, as I've just said. Tautomeric shifts are rare, but they may be of importance in DNA metabolism because some alter the pairing potential of the bases. Watson and Crick pointed out that the structures of the bases in DNA are not static. Hydrogen atoms can move from one position in the purine or pyrimidine to another position. For example, from amino group to a ring nitrogen, okay? That is from number 1 position to number 6 position of the purines. And this is observed even in the case of pyrimidines, where the hydrogen shifts from the number 3 position to number 4 position of the pyrimidines. This results in tautomeric forms, different forms as compared to the more common forms. So, all the tautomeric shifts are rare, as I've said. They may be of considerable importance in DNA metabolism because some of them alter the pairing potential of the bases. All DNA bases, that is adenine, guanine, cytosine, thymine, they have unusual tautomeric forms, which are, however, rare in occurrence. The nucleotide structures that are the common ones, they are more stable forms. So, the keto form of thymine, amino form of cytosine, amino form of adenine, keto form of guanine, these are all common forms and they are more stable as compared to their tautomers, that is the enol forms and the imino forms. So the more stable keto forms of thymine and guanine may infrequently undergo tautomeric shifts to less stable enol forms, and more stable amino forms of adenine and cytosine may undergo tautomeric shifts to less stable imino forms. The bases would be expected to remain in these rare forms for a very short period of time, as they are highly unstable. They are highly unstable. But if a base existed in the rare form at the time of replication, and if it gets incorporated into the nascent DNA, then there is a problem. A mutation would result. When the bases are present in their rare imino or enol form, they exhibit unusual pairing behaviors. Adenine pairs with cytosine instead of thymine, and adenine pairs with cytosine instead of thymine, and guanine pairs with thymine instead of cytosine. So the net effect of such mismatched base pairing is that AT is converted or is changed to a GC base pair substitution, or a GC base pair is changed to a AT base pair substitution. The mutations resulting from tautomeric shifts in the bases of DNA involve the replacement of a purine in one strand of DNA with the other purine, and the replacement of pyrimidine in the complementary strand with the other pyrimidine transitions. Pyrimidine. These are called as the transitions. Watson and Crick in 1955 have hypothesized that the occurrence of the bases in tautomeric forms possibly provides a mechanism of for mutation during DNA replication. If in an old strand of DNA, guanine is in its rare enol tautomeric condition, it will pair with normal thymine. It will pair with normal thymine instead of cytosine in the new complementary strand during DNA replication. This may result in a new DNA molecule containing exceptional base pair, that is guanine base paired with thymine. Guanine base paired with thymine. The tautomeric shift is not stable, and in the next replication, guanine will go back to its normal state and it will pair with cytosine, and the thymine which was added in the complementary strand pairs with adenine, and this results in a new base pair as compared to the parental DNA, that is AT base pair instead of a GC base pair. So, two kinds of DNA molecules are formed, some with normal base sequences and some with changed base sequences. The two kinds of DNA molecules formed by transition will eventually be separated into different cells, which may result in recognizable mutations. Frameshift mutations, insertions, deletions of one or few base pairs within the coding regions of the genes result in an altered sequence of amino acids at the translation of the codons, and this is known as frame shift mutations. In the nucleic acid, the nucleotides may be read in groups of non-overlapping consecutive triplets or codons referred to altogether as the reading frame. During translation, codons in the reading frame are translated into specific amino acids. Thus, if an insertion or deletion of the nucleotide occurs, this could result in the alteration of the reading frame. It completely changes the amino acid sequence. Such mutations are known as frame shift mutations, as is observed over here. In the wild type, this is the reading frame in the mRNA, which has been transcribed from this DNA. AUG, which will code for methionine, that forms codon 1. Then AAA, codon 2, codes for lysine. GGG, codon 3, codes for glycine. But as a result of a CG base pair addition, there is a change in the reading frame from C onwards in this mRNA. So codon 3, instead of coding for glycine, it codes for arginine. And then codon 4 is coding for proline in the wild type, but it codes for alanine in the mutant as a result of this frame shift mutation. The addition or deletion of nucleotides in the multiples of 3, however, will not alter the reading frame. Thus, the protein in such cases would likely have either an extra amino acid or an amino acid might go missing. Now, this is frame shift mutations are caused by mutational errors during the DNA error repair or replication. They can also occur by exposure to acridine dyes, which are capable of introducing frame shift mutations. The resultant codons after frame shift mutations can be of 3 types. They can be sense codons, missense codons, or nonsense codons. In the case of sense codons, these are codons that are read in the same manner as before the frame shift mutation. Missense codons will result in different amino acid, an incorrect amino acid in the polypeptide chain. Nonsense codons is a stop codon for which there is no corresponding tRNA, and it results in the termination of the translation process. So, as a result, a short length polypeptide chain may be formed. Hence, frame shift mutations result in an abnormal or defective protein product containing an improper sequence of amino acids. These are the references, web links. Thank you.